efficacy of 87 as well as 60. So it indicates that this tetra bis dimethoxy is showing lower range of encapsulation efficacy than those tetra dimethoxy. Well, we checked the antibacterial too. We found that when it comes to vesicle entrapped form, tetra-bis dimethoxy has shown MIC far better than tetra-dimethoxy. So we can see that though entrapment was less, still the efficacy is higher. Then we went to check the functional compatibility. So this all we have done before going to 3D printing. That is a systematic process was performed to check everything and then go for a hybrid formulation. So we did an functional compatibility test and we found that when it comes to spectral identification of curcumide, that benzene ring was obtained around 1500, and that confirmed that the entrapped vesicles still hold, like structural conformity was obtained from there. Then we went to check the morphology. Now this morphology was performed, morphology was tested using FESAM, not TEM. It was because non-availability of TEM. So we developed a method, and using that method, we were successfully able to check the liposomal morphology using FESAM. So figure A, B, and C are from confocal laser microscopy, and figure D and E from FESAM. So that confocal microscopy is a bit different process than FESAM. For confocal microscopy, a dye was labeled within the vesicles and then it was tested. While in FESAM, the vesicles were processed through a series of process where crystal violets was used to treat, and then after doing that, gold coating was done and we found a good spherical structure of vesicles. Then we tested the physical property and we found that the composition what we have optimized had bit lower in zeta potential than the lower composition of phosphatidylcholine, while the size as well as PDI also differed in when it comes to composition of phosphatidylcholine. So still the composition was varied, because when we went to use 50, that is the initial composition of phosphatidylcholine, the entrapment efficacy was less, and then we have to improve the concentration and find out an reliable entrapment efficacy that can be further processed. So after doing this, we went to check the deformability. Deformability was again performed using an improvised method where we took an specific filters and we tried to transfer, and before filtration and after filtration we checked the size and we found insignificant change in size, and that defined that the flexibility or flexible nature of the vesicles what prepared. So that were all the physico-chemical properties we have been tested. Now we came to test the entrapment, sorry, presence of those lipid entrapped composition which is going to applied over skin will enter the skin and able to penetrate the keratinized cell or not. So first we label the phosphatidylcholine based vesicles with propodium iodide. Now this dye can penetrate inside the cell and reach to the DNA when cells are dyed. So if our formulations, which is enough successful in lipoidal nature, it should allow the entrapment in a live cell. And we found so when it comes to the checking the efficacy, we tested over the HiCat cell. We grow the HiCat cell in 6-well culture plate and at IC50. So those calculation we will be seeing later on the slide. So we we found the IC50 concentration and we treated the cell at those concentration, and after treating, the slides were prepared and we used the uh, fluorescent microscope with specific emission and excitation wavelength of light, and we found uh, the dye, propadium iodide dye based formulation has already penetrated inside the cell. So you can see in both mode, when it comes to cell which were treated without propadium iodide loaded vesicles, as well as when it was treated with added vesicles. Further, we tested using flow cytometer. This is another way where we can test again the presence of dye using laser beam based operated flow cytometers. So we found that both, that is tetra dimethoxy and tetra bis dimethoxy entrapped, showing the dye intensity, while when it comes to the lipid where no dye was added, the intensity was very less, and which might be probably because of noise peak. So that confirmed that yes, the preparation can reach within the cell. So this is the biocompatibility we did, and based on this, we performed the previous studies. That is the whether cell penetration can be seen or not. So you can see here, that is the biocompatibility has been performed over macrophage as well as on keratinite cell. So we found that at concentration something around 125 over the in macrophage, as well as when it comes to keratinite cell, the concentration something around 15.6 microgram for tetradimethoxy is compatible, while when it comes to bis dimethoxy, uh, the keratinite cells are compatible around 3.9. So those are very low, and this is very good sign that when we require a lower dose to get the effective results. So at those dose, we try to check the efficacy, whether inflammatory response can be seen or not. That is, the reduction of nitric oxide was tested using Grice reagent. And we found that when it comes to raw and when it comes to entrapped, equivalent weight, both are showing similar kind of results. While the blue bar graph, you can see that at those tested concentration, cells were still alive. That is, the viability was still maintained, what we have checked previously. So this confirmed that both either in entrapped form or in native form, they can reduce the inflammation. Now we will compare those two results with further studies what has been done in for dimethoxy and bis dimethoxy. So these results were for tetra dimethoxy and tetra bis dimethoxy, and we will see the other two metabolites, how they operate. So when it comes to reduction in inflammation, at 13 microgram raw dimethoxy was effective, and when it comes to vesicular entrapped, it has reduced. That is, the dose has been reduced, but still effective dose we took as 13 only. That is, as 13 microgram per mil, it can reduce the nitric oxide secretion. While when it comes to bis dimethoxy, almost to zero. That is similar to control cells, the results was obtained. So we can see that tetra, when it comes to tetra dimethoxy and tetra bis dimethoxy, the dose are bit lower for dimethoxy and bis dimethoxy. And when it comes to curcumin, the dose are higher for tetrahydro, which is most compatible form. That is because of off-white in color, and it does not require preservation from light. But the dose at 15, it was able to reduce something around 47%. But when it comes to curcumin, at 15 microgram, it could able to present the result as similar to the control. So this define that these two metabolites are highly effective if it comes to these two, and the other one, that is tetrahydro. But it depend on the availability what we can prepare. But this is an comparative results what we have already presented and published in good high impact factor journals, that is biological macromolecule and nanomedicines. So next we check. This is the further efficacy we have checked in the form of compatibility for the TDM, that is tetra dimethoxy. And so the test what we have performed here was further tested in the form of compatibility, and we found that in presence of LPS2, the cells are viable. Like LPS, sometime if we treat high concentration of LPS to induce inflammation, it can cause the death of cell too. So we have to check that too, that the tested LPS concentration does not affect the cell viability. So ultimately aim was the reduction in inflammation should come from the viable cell, not from the dead cell. So after doing this, we tested the that is the diffusion study using membrane, which is having molecular weight cutoff of this, and we found that both are showing good release comparative to the entrapment efficacy. And finally, we tested the stability before we moved to the 3D printing, and we found that stability is little reducing. So from here, we decided that we will go for the printing of this vesicle entrapped preparation in the form of composite using a gel printer. So first we went to the software, that is CAD, where we developed or we design what kind of printing we need to do. So after designing in the form of CAD, we exported those design to the 3D printer, and then we went to printing. But before doing this, we first analyze the mechanic of fluids, that is the mechanics of fluid we have to understand before we go and print it. So we took the composition as I have presented in the beginning, that polyvinyl as well as with different polymeric composition, we prepared the gel, and in those gel we transfer these vesicles and trapped formulations, and then we printed it. So after printing, we tested for further physico mechanical properties, that is whether it is useful or not in term of in term of dressings. But this is the preliminary. The compatibility we need to test whether those rings of curcuminides are still present or not after entrapping. So we can see those rings were masked, and when it comes to when it comes to liposomal preparation, were reduced. But when it comes to dried form of composite, it shows reappearing of this specific rings related to the curcuminoids, and that confirmed that the stable and entrapped form of vesicles are present in the composite. Then further we tested the compatibility using DSC, which does not shows any change in the endothermic and exothermic curve. So we found that it's good to have such preparation. Then we tested the morphology. So when we comes to check the morphology from the surface and from the cross section, it is showing in the figure B, you can see that void-like structures are available, and those void-like structures are the vesicles which is present inside the polymeric composite. Further, we tested the contact angle and we found that the range of contact angles are highly acceptable for dressing which can be applied over the wounds. Now, important thing is we need to go and check again whether it can scavenge the process which is going to be happen during the healing of wound, as well as whether it can show viability and inflammation reduction or not. So we tested everything and we found that viability is perfect for the tested size of composite, as well as when it comes to, okay, I didn't add this slide of the inflammation, but we took a specific size of composite and we dissolved those composite in DMEM, that is the culture media, and those elutes were tested for reduction, whether it can show reduction in inflammation or not. So we found that it can still reduce, and at last we went to check whether it can show efficacy towards wound healing or not. So we tested this in the form of cell migration assay, and we found that these 3D printed composite, which was fortified with vesicle and trapped curcuminoids, was effective. And this we will see in the form of comparison, that is dimethoxy, that is film, which is entrapped with vesicle entrapped tetradimethoxy, and film which is fortified with tetra bis dimethoxy. So we found that after 48 hours, dimethoxy in the form of film and trap showing almost 90% of cell migration, and bit less was presented by the film which was fortified with vesicle and trapped. So these results were further compared with the other two metabolites, that is dimethoxy and bis dimethoxy. So we can find here that both in the form of dimethoxy and bis dimethoxy, when it comes to film and trapped form, it is showing similar result, that is more than 80% of cell migrations. And when it comes to tetrahydro and curcumin, we can see that tetrahydro is showing 96% of cell migration, while curcumin, which was entrapped in the film, showed 87% of cell migration. So we can see there is a bit of variations, but still all are showing a good results. That is all for metabolites. Sorry, 5 metabolites and of curcumin, that is curcuminoids are effective in this process. So these studies conclude that these preparations in the form of vesicles showing instability after some specific time of storage, so needs to be transferred in the third form, that is the hybrid form of the preparations. And this 3D printing technology is showing efficacy in this form, and we can improve those preparation stability when using this technology of 3D printing. So I would like to thanks, and yeah, if we have any suggestions or questions, I'm happy to answer you.
Now I request today's chairpersons, Professor Subhash C. Rinda and Professor Mehul N. Patil, to present the views on this session.
Thank you, madam. It was a nice session, and it is on latest 3D printing used in different formulation development, and so it was he has given delivered a very good outlook to this presentation. A lot of learning. I think our audience or those who are with us in online, they might have learned many things which can be utilized, and this is a highlighted area in present research area, and a lot of things can be done on these particular aspects 3D printing. So I would like to request from the audience any questions, so thereby he can explain. Hello. Hello. Any questions from the audience? Any question from the organizers?
Good morning, sir. Thank you so much for your elaborative session. Myself, Biola from 4th B Farm. I have small question, sir.
Yes.
If we have to select any natural component for establishing its activity, what are the aspects we have to consider? Because you have used so many naturals like turmeric, eucalyptus, and guava, etc.
We work on different aspects. So selection of such compounds depend on your what is objective. What we our objective here is to reduce the waste and use the component which is available in the form of waste. Eucalyptus is highly available here from the paper industry. They use the stem and bark and leave the leaves in the form of waste. So we tried to get something from those waste. Then when it comes to rhodomatone, which is coming from Rhodomytus tomentosa, it is an edible fruit, which is coming from plant which known as Rhodomytus tomentosa. So has been already stabilized everything, that is PKPD, everything has been done, and that indicate the potency of those compounds. Now, when it comes to curcumin, it's depend on what we want to do. Like curcumin, still we can see several research are published, but when it comes to metabolites, you have to first consider the thing is whether it is available with you or not, whether it is easily available with you or not, or if you want to procure from somewhere, it can be in your budget or not. So it's not doing research in a way where you will go and find out something highly effective, but which is not available to you easily. So there are, in my opinion, the first thing what I will tell you is that what is your objective? Fix your objective depending on the availability, and then not only availability of those compound, but also the availability of instrumental analysis what you can perform. Setting a very high objective may sometime create time loss, and being a scholar, we don't have much time to go and spend on several things. So natural products are nothing like new. We are already growing, and we know from the Indian Ayurveda, most of synthetic products are analog of those natural product based structures. So when we know their efficacy, we can go in those direction. If we don't know their efficacy, blindly going with natural products is just waste of time and economy. This what I can say. Like selection for our lab is basically based on valorization of waste product. So we look where we can find a material that can be used for in a cost-effective form.
Thank you, sir. I was very much inspired with your work, sir.
I would like to request Mehul Patel sir to say a few comments.
Yeah, good morning, sir.
Good
Uh,
morning.
Good
yeah,
afternoon, sir.
yeah, good afternoon. So this is a really 3D printing is a technology, and sir has wonderfully presented how the 3D printing can be utilized in our pharmaceutical sectors. Because in futures, this will be the one of the new technologies where actually we are facing so many limitation with the existing one, and that can be possible that overcome by means of 3D printing technologies.
Yes.
So wonderful. Students may come to know that because recently one of my students also work on that projects, and then we come to know that how this technology is beneficial in drug delivery systems. Sir, I want to just discuss one points on that aspects regarding the materials that I think you have used. Even we have also used the same kind of materials. So, what are the actual requirements? Because the filament is the main components in 3D printing technologies. So, is there any
So
quite specific or something requirement is there that these materials
no,
only can
the
be
filament-based 3D printer is specific filaments you need to go and buy.
okay?
3D printing technology have various categorizations. Among that, FDM is one of them.
Yeah.
So for FDM, it may be not as economic as gel printer, but
Yes.
advantage with filament based is uh, you can design in a form that can give a good dynamic structures.
Okay.
So when it comes to filaments, usually we purchase ready to use, where
Okay.
drugs are most of case drugs are already added.
Okay.
So that is an customized form of filament we purchase. But when it comes to gel based printer, we have to play with the fluid mechanics. Until
Okay.
we cannot optimize those fluid mechanics, it is not easy to print them.
Yeah, and that is what challenge is there with
Yeah.
the 3D printing.
Definitely, these
Yes.
are many outcomes are coming out of this from this area of research. I've seen they are trying for the natural polymers and designing sustain delivery and all, and to critical drugs and all. I have seen a lot of works are going on throughout our country in most of the labs. So I hope this is a very highlighted area, and we'll proceed. Much more we can concentrate on this area to do something for the solving the healthcare problems. So thank you, sir, for a nice presentation. So
Thank
I
you,
hand over
sir.
to the organizers to take care.
Thank you.
Thank you, Dr. Sudarshan Singh, for a nice and informative lecture. And thank you for the advice of both the expert. So hope all the discussion will remain fruitful and students will be able to take the fruitful ideas from here to innovate their projects. So now we move to our next session. Uh, uh, I asked the hosting college to introduce the next speaker and carry out the next lecture. Thank you.
Sir,
Yeah.
pardon, Rajiv there, sir. So we have a inspection ahead. I have to do a lot of things.
Okay.
Little
Okay,
bit.
there is no issue.
Thank you. Thank you very much.
Yeah.
Thank
Thank
you
you
all
very much,
for
sir.
giving me this opportunity. I'll meet
Mhm.
next in some
Thank
events.
you.
Thank you.
Hmm.
Yeah.
Thank you, sir, for addressing the all queries so clearly and effectively. Uh, for that highly informative and insightful presentation. Your valuable inputs have greatly enhanced our understanding of this importance of area in pharmaceutical sciences. Now, as a token of appreciation for sharing your valuable knowledge with us today, we would like to share present a certificate of appreciation to our esteemed speaker, to Dr. Sudarshan Singh garu. Thank you, sir. We now move on to the next expert talk of this session. To introduce the next speaker, I request Mrs. G. Krupamayi ma'am to kindly present the profile of Dr. M. Vamsi Krishna sir.
Respected dignitaries, esteemed faculty members, and dear participants, a very good morning to everyone. Myself, G. Krupamayi, Assistant Professor, Department of Pharmaceutical Chemistry, Vijay Institute of Pharmaceutical Sciences for Women. I feel elated and privileged to introduce the profile of our distinguished speaker, Dr. M. Vamsi Krishna sir, for this scientific session. Dr. Vamsi sir has completed his B.Pharm, M.Pharm in the Pharmaceutical Analysis Specialization, and PhD from Andhra University campus, Vishakhapatnam. Sir has demonstrated remarkable academic excellence, having qualified in the GATE examination with an impressive 97.84 percentile, securing All India rank 133, and also achieved state third rank in ESET 1999 organized for pharmacy diploma holders. Sir is the recipient of several prestigious recognitions, including the Dr. P.D. Sethi Award at the 59th Indian Pharmaceutical Congress held at Banaras Hindu University, Varanasi, and the Sri J.G. Rao Award at the 78th Convocation of Andhra University, Vishakhapatnam. In addition, Sir has received 4 Best Research and Technical Paper Awards at national and international conferences. Academically, Sir has made significant research contributions with more than 60 publications in reputed national and international journals. Sir has also authored 2 book chapters on advanced sample preparation techniques in analytical toxicology and their applications in LCMS-based metabolite profiling. His expertise is further recognized through his role as a reviewer for reputed international journals, including the Journal of Separation Science, Biomedical Chromatography, and the Journal of Molecular Structure. With over 17 years of rich experience in teaching, research, and industry in India and abroad, and Sir has extended his expertise as a scientist at the R&D Center of SMS Pharmaceuticals Limited, Hyderabad, and is currently working as Deputy Manager in the Peptides Analytical Division at Energon Labs, Hyderabad. With his vast experience and expertise in pharmaceutical analysis and analytical sciences, we are honored to have such an eminent personality with us today to share his knowledge and expertise insights with us. With this brief introduction, now I welcome and respectfully invite Sir to take over the session. Thank you, sir. Sir.
Whether he has joined the session? Hello. Krupamayi. Hello.
Madam, your voice is not audible.
Yes, ma'am.
వంశీ కృష్ణ సార్ స్క్రీన్ షేర్ చేశారా?
చేస్తాను మేడం, చేస్తున్నాను మేడం.
ఓకే సార్.
నాది పవర్ పాయింట్. డెస్క్టాప్ విండో. స్క్రీన్ ఎలా?
Sir,
Madam. Just I am sharing the PowerPoint, madam. Right. Input hide. Put new sheet. Madam, is it visible?
Yes,
your
sir,
slides
visible.
sir, it is visible, sir.
Okay, madam.
In presentation mode.
Okay, madam. Okay, very good morning to all. Myself, I am Dr. Vamsi Krishna. I'm working as Deputy Manager in the Energon Labs. I'm working in the area of peptides. Is it okay, madam?
షో మోడ్ లోనే ఉంది మాట్లాడితే మీది కనిపించట్లేదు ఇక్కడ. సరే మేడం. ఓకే ఓకే. Archana
Sir,
madam.
put on the slideshow mode.
Slideshow mode. Slideshow.
అది
On the top of the screen.
Archana madam కి షేర్ అని చూడు.
Archana madam. Slideshow.
This
Slideshow. This one. Sir, is it okay?
నీ వాట్సాప్ లో ఎక్కడ నా పంపి. Archana madam
Try to move the slide.
Yes.
Go on second slide.
I am moving.
No, but it's still your first slide is showing.
Okay. 1 minute, I am sharing my to WhatsApp.
Share. Sir, share. అట్లా వాట్సాప్
ఇది నేను స్లైడ్ వ్యూ వెళ్ళా పెద్దది.
ఇట్లా చెప్పేసేయాలి.
ఇది మూవ్ అవటల్లా.
Sir, click on slideshow. Click on
slideshow.
Sir, you can start the presentation.
Yes, it is okay. You can start. Please unmute yourself.
Yes. Yeah. Very good morning to all. So myself, I am Dr. Vamsi Krishna. I am working as Deputy Manager in the Energon Labs. I am working on peptides. So today I am going to give a presentation on applications of CE and CEMS in the biopharmaceutical process development and their quality control. Can I have next slide? Yeah. So coming to the biopharmaceuticals, these biopharmaceuticals also referred to as biologics. These represents a group of or a class of therapeutic agents which we are deriving from biological systems, and these are generally developed through advanced protein engineering. coming to the class comprises these biological molecules contain these wide range of molecules, including recombinant proteins, oligonucleotides, peptides, nucleic acids, and cell-based therapies. All these are comes under these biopharmaceuticals. Now, if you see the differences, key differences between small molecules and biopharmaceuticals, if you see the small drug molecules, their molecular weight is very low compared to the biopharmaceuticals. If you see the molecular mass of small drug molecules, this is generally less than 0.5 kilodaltons. And in the case of biopharmaceuticals, the molecular weight, molecular mass is greater than 4 kilodaltons. These are very large range. And coming to the structure, these small drug molecules are very simple and well-defined structure is there. But in the case of biopharmaceuticals, they are very complex and heterogeneous. The structure is very complex and heterogeneous. And comes to the source of these small drug molecules and biopharmaceuticals. Generally, these small drug molecules we are deriving by chemical synthesis mode. But in the case of biopharmaceuticals, we are deriving these from live cells and organisms via protein engineering. Coming to the stability aspect, these small drug molecules are very stable. Coming to the biopharmaceuticals, these are generally unstable and these are very sensitive to the external conditions by means of temperature, pH, shear, very small variations in the conditions. They are very sensitive and their stability is going to be altered. And also, in the case of small drug molecules, these are generally non-immunogenic. We are not going to observe any immunogenic reactions in the case of small drug molecules. But in the case of biopharmaceuticals, these are mostly immunogenic. They are going to produce immunogenic reactions. So these are what the key differences between small drug molecules and biopharmaceuticals. Coming to the next slide. Yeah, coming to the structure, these already we discussed. The biopharmaceuticals are very complex and these are very sensitive to the environmental factors. That's why biopharmaceuticals required specialized care during development, during production, and during the storage and transportation. Specialized care is required for the biopharmaceutical. And in contrast to small molecule drugs, biopharmaceuticals are characterized by high molecular weight, complex, and heterogeneous structure. And these biologics are more susceptible to degradation, and these are producing immunogenic response. And there are more stability concerns compared to the small drug molecules. Coming to the next slide. Yeah. So, despite the remarkable advancements in biopharmaceutical development, so these have very structural complexity, and these biopharmaceuticals are having physicochemical heterogeneity. So, due to this complex structure and physicochemical heterogeneity, we require sophisticated analytical characterization. We require a sophisticated analytical instruments, and we require a characterization and also very diverse techniques are required in the quality control methodologies. So ensuring the consistency, batch to batch consistency, efficacy, and safety of these biological molecules, it requires a, for testing of these, it requires a very sensitive and robust analytical technologies are there because these biopharmaceuticals are very complex and very heterogeneous. If there is a small variation in their physicochemical characteristics, so their biological responses are going to be altered. So that's why for ensuring the consistency, for ensuring the efficacy and safety, we require very sensitive analytical methodologies and robust analytical methodologies are required. Unlike small drug molecules, uh, so if you see the small drug molecules, generally small drug molecules are we are going to be characterized by means of routine chromatography techniques like HPLC and GC. We are characterizing the small drug molecules. But in the case of biologics, simply one or two techniques are not enough. We need a integrated approach combining multiple orthogonal analytical technologies. We will see in the future slides. So we require multiple techniques, multiple orthogonal analytical techniques are required. So coming to the rigorous characterization, so for these biopharmaceuticals, rigorous characterization is critical for both originator biologics and biosimilars. Here there is another term that is what called biosimilars. So the original innovator molecules we are calling as biologics, and after expiry of patents, people will develop a biosimilars. So these biosimilars are biological products. Those are very similar to the approved reference biologics. So these biosimilars are very similar to the biologics, and there is no clinical meaningful difference in the case of safety, purity, and potency. These are very much similar to the originator biologics. So those are what called biosimilars. So now our markets flooded with these biosimilars in the future we are going to see. So that's why there is a rigorous characterization analytical techniques are required to monitor the physicochemical and structural characterization of biosimilars. As patents of original biologics expire, biosimilars present an opportunity to expand patient access to biologic therapies while reducing the healthcare costs. So, due to the advent of these biosimilars, so but the products are very accessible to the patients, and also the cost also reduced in the case of biosimilars. And regulatory agencies like US FDA, US Food and Drug Administration, and EMA, European Medicine Agency, have established stringent guidelines for biosimilars. So people are developing these biosimilars, and for approval of these biosimilar products, regulatory authorities like US FDA and EMA providing a stringent guidelines, and there is a need for meeting these stringent guidelines. We need a robust analytical results. Robust analytical frameworks are required to detect potential variations. What are the variations between original biologic and biosimilar? And how you are characterizing your biosimilar with the help of a robust analytical techniques. So these are all required to detect the potential variations in the biosimilars that could impact the therapeutic performance, because small variation in biosimilar, it will affect the therapeutic performance of the biosimilar. Please next slide. Yeah, these are what the group of some of the examples of biopharmaceuticals, namely recombinant proteins, monoclonal antibodies, vaccines, gene therapy products, cell therapy products, and tissue engineering products. All these are different types of biopharmaceuticals. And generally, these we are producing recombinant proteins, monoclonal antibodies, vaccines, we are producing by means of a recombinant DNA technology. And we are going to use E. coli recombinant DNA technology and E. coli recombinant DNA technology in Chinese hamster ovary cell lines. We are using and different microorganisms we are using, and we are producing these biopharmaceuticals. Please next slide. Yeah, what are the current challenges in biopharmaceutical analysis? What are the challenges in this biopharmaceutical analysis? So, if you see the biopharmaceutical products, those are having diversity in the molecular structures. Their molecular structure is very diverse. So, due to this structural complexity and inherent heterogeneity presents significant analytical challenges. So, it is going to impact these structural complexity and heterogeneity will impact the characterization and regulatory compliance. So this heterogeneity, this will arise from multiple factors, including variations in the molecular size. If simply variations in the size and charge, so these are all, and glycosylation, all these are going to be influence the therapeutic efficacy of your products. So moreover, these protein based biopharmaceuticals exhibit intricate folding patterns. They have very complex folding patterns and a higher order structure. So due to this, secondary, tertiary, and quaternary conformations, we have a higher order structure of biopharmaceuticals. So these further gives complexity to our analysis. So this complexity stems from batch to batch variability inherent to the production. So in the case of a production of these biopharmaceuticals or biosimilars, so there may be a batch to batch variability because these we are producing by means of a diverse expression systems. So diverse microorganisms we are going to use, diverse cell cultures we are going to use. So due to this, we are going to observe a batch to batch variability, and there is a chance of various post translational modifications, PTMs, such as glycosylation in the case of monoclonal antibodies, and disulfide bond formation, for example, in the case of insulin, and uh, uh, various structural variations we are going to be observed in the case of these post translational modifications. Please, next slide, please. So, what are the various methods we are going to adopt for the quality control of these biopharmaceuticals? So, what are the various key attributes we are testing in the quality control? So, analytical methods applied to characterization of these biopharmaceuticals, if we see, several analytical methods have been employed to assess the biopharmaceuticals key attributes. So what are these key attributes we are testing in the quality control is uh, identity we are testing, purity, potency, structural integrity, aggregation, stability, glycosylation, post-translational modifications. All these key attributes we are testing in the quality routine quality control, and then we are releasing the biopharmaceuticals. If you see that in the case of a first one, identity testing. So for the identity testing, so what are the various analytical techniques we are going to use in quality control? So one is MS method. So MS method, generally we are going to use a mass spectrometric method. These mass spectrometric methods we are using to determine the key features like molecular weight and sequencing with high precision. So peptide sequencing we are determining, and we are determining the molecular weight by means of a mass spectrometry. So for this identity testing, we are using mass spectrometric methods, and we are using peptide mapping. So we are also performing peptide mapping for the identity testing of biosimilars or biopharmaceuticals. So for this peptide mapping, we are going to adapt enzymatic digestion method. So for this enzymatic digestion method, we are going to use a trypsin digestion we are using, and then we are breaking the bonds, and we are determining the peptide chains, peptide mapping we are determining, and we are determining the primary structure of our proteins, our biologics, our biopharmaceuticals. This peptide mapping is a qualitative aspect. And then we are using NMR spectroscopies also for providing information on molecular structure and dynamics of these biopharmaceuticals. So this is what identity testing. Then coming to the purity, quantity, and impurity analysis. These are what another analytical purpose we are measuring. We are key attributes we are measuring: purity, quantity, and impurity analysis. So for these applications, we are going to use the techniques like HPLC high performance liquid chromatography, and CE, capillary electrophoresis, CE and CEMS, capillary electrophoresis and mass spectrometry combination we are going to use. And then conventional method like SDS phase, sodium radical sulfate polyacrylamide, gel electrophoresis method, we are going to use SDS phase. And we are using biosensors like immunosensors we are using, and the technique like invisible spectroscopy absorption at 260 nanometers and 280 nanometers we are generally using for determining this purity, quantity, and impurity analysis. If you see the HPLC, in the case of HPLC, what we are going to do is the key features, separate the components based on interactions with the stationary phase. So, basing upon the interactions with the stationary phase, we are separating and we are determining the purity and quantity. So, quantitative analysis we are performing by HPLC. And in the case of SE and CEMS, here we are separating molecules. We are separating the molecules based upon their charge to size to charge ratio, basing upon the size of the your molecules and basing upon the charge of ions, we are separating the molecules in the electric field, and then we are performing the quantitative analysis. Then routinely SDS phase we are using. These are very laborious techniques. This is a conventional technique, and it separates proteins based on molecular weight under denaturing condition. So this technique is useful for qualitative and semi-quantitative assays. And biosensors like immunosensors we are using to detect specific interaction between biomolecules, and we are going to monitor the real-time binding events of biopharmaceuticals. And then coming to the UV visible spectroscopy. So here we are measure the protein concentration based on their intrinsic light absorption, generally at 260 nanometer and 280 nanometer. This is a quantitative aspect. Please, next slide, please. Yeah. Other key attributes like potency, quantity, and bioactivity. So the potency, quantity, and bioactivity, we are going to determine the techniques like ELISA, enzyme-linked immunosorbent assay we are going to use, and various electrochemical and optical biosensors we are going to use, and cell-based assays we are going to use, and surface plasma resonance spectroscopy, resonance SPR technique we are going to use. Coming to the ELISA, so here we detect and quantifies specific proteins or antibodies using antigen antibody interaction. The basic feature is antigen antibody interactions we are performing and we are determining quantitatively. And then electrochemical and optical biosensors, we are measuring the biological interactions through changes in electrical optical signals. By changes in electrical or optical signals, we are determining these biological interactions. And then by means of cell-based assays, we are going to evaluate biological response of living cells to a biopharmaceutical. How the cells will respond to a biopharmaceuticals, we are going to analyze by means of a cell-based assays. And surface plasma resonance we are going to use to monitor the by real-time binding interactions. This is a quantitative assay. Next slide, please. Yeah. So another key important attribute is structural characterization. Structural characterization we are performing by means of XRD, X-ray crystallography we are going to use, and other technique is called circular dichroism and FTIR. So these are what the three important techniques: X-ray crystallography, circular dichroism, and Fourier transform infrared spectroscopy. These three techniques we are going to use for structural characterization of biopharmaceuticals. And in the case of X-ray crystallography, we are determining 3D structural information. We are detecting the 3D structural information by using XRD spectroscopy. And then in the case of circular dichroism, we are assessing, in the case of a circular dichroism, we are assessing the protein secondary structure. Protein secondary structure coming from alpha helix and beta sheets of biopharmaceuticals. We are determining that secondary structure by means of a circular dichroism technique. And in the case of a FTIR, by means of FTIR, we are also analyzing the secondary structure. And here we are observing amide bonds. In the case of a FTIR spectrum, we are observing the amide bonds of our biopharmaceuticals. So, by means of this FTIR and CD, we are determining the secondary structure of our proteins or biopharmaceuticals. So those are what the analytical techniques which we are using for structural characterization. Now coming to the another important key attribute characterization is aggregation, stability, and degradation. So these are what the key parameters, we are key attributes we are going to analyze by means of analytical techniques or aggregation. So due to this aggregation, so our biopharmaceutical activity is going to be, efficacy is going to be reduced, and sometimes we are going to observe due to aggregations, immunogenic responses, unwanted immunogenic reactions, we are going to observe. So due to that, we are going to we are going to determine the aggregation of biopharmaceuticals, and we are going to determine the stability of biopharmaceuticals, and we are performing stable degradation studies, and then we can establish the degradation patterns of biopharmaceuticals. And for these aggregation, stability, and degradation studies, we are going to use the technique called dynamic light scattering technique, DLS, that is what called. So by means of this dynamic light scattering technique, we are going to measure the particle size distribution. We are measuring the particle size distribution in solution form by analyzing light scattering pattern. So by means of the light scattering patterns in the DLS, we are going to measure the particle size distributions in the solution form. So that's what the technique DLS we are going to use for aggregations. Then another important technique is size exclusion chromatography, multi-angle light scattering technique, MALS, SES MALS, that is what called size exclusion chromatography and multi-angle light scattering technique. By means of this size exclusion multi-angle light scattering technique, so we are separate the molecules based upon the size with the help of a size exclusion chromatography. So with the help of a CC, we are separating the molecules based upon size, and then by with the help of MALS, multi-angle light scattering, it provides molecular weight. So we are going to determine the molecular weight with the help of a MALS. So we are separating sizes with the help of a size exclusion chromatography, and then we are determining absolute molecular weight of our molecules with the help of a multi-angle light scattering technique. Then ultracentrifugation technique. Ultracentrifugation technique, by means of analytical ultracentrifugation technique, we are going to measure the sedimentation behavior of our macromolecules. So these biopharmaceuticals are biosimilars or big molecules, macromolecules. So with the help of analytical ultracentrifugation technique, we are going to measure the sedimentation behavior under high centrifugal force. So by means of that, we are going to determine the aggregation behavior of our biopharmaceuticals. We are going to determine. And another important simple technique is DSC, differential scanning calorimetry technique. And with the help of this DSC, we are going to measure the heat changes. So with the heat, it measures the heat changes associated with protein unfolding. So with the heat, how protein is going, how our biopharmaceutical are going to be unfold. So here in the DSC, we are applying a heat. We are applying a heat. So with the heat, how the um, unfolding behavior of this uh, biopharmaceutical we are going to measure with the help of a DSC. And then LCHRMS, so liquid chromatography and HRMS, high resolution mass spectrometers, mass spectrometry techniques like QTOF, quarter pole time of flight, or RB trap technique, we are going to use for determination of these aggregates. So here we are combining separation capabilities like size exclusion chromatography we are going to perform, and then we are performing the mass analysis of aggregates with the help of a QTOF, quarter pole time of flight mass spectrometer. Next slide please. Yeah, so another important key attribute we are going to measure is glycosylation and post-translational modifications. So this glycosylation is very important. The biopharmaceutical activity efficacy is depends upon the glycosylation. So we are going to determine this glycosylation, and we are going to determine post-translational modifications. Because of these glycosylation and post-translational modifications, efficacy is also going to be altered, safety is going to be altered. So for determining this glycosylation and post-translational modifications, we are going to use techniques like LCMS is one technique, and then helic MS. This is one type of technique. Hydrophilic interaction liquid chromatography, hydrophilic interaction liquid chromatography, and MS mass spectrometry we are going to use for determination of these PTMs and glycosylation. And then next generation sequencing technique we are going to use for sequence of DNA and RNA fragments. If you see this LCMS, so we are going to perform detailed analysis of glycan structures. So complete glycan structures we are going to be determined, and post-translational modifications we are going to determine by means of a LCMS technique. And then in the case of a helic MS separates glycans based upon hydrophilicity. In the helic technique, helic, that is a liquid chromatography technique, helic technique, by means of helic technique, we are going to separate the glycans. So we are going to separate the oligosaccharides. We are going to separate the glycans based upon their hydrophilicity. And then coupling with mass spectrometry, we are going to get the clear structural information of these glycans. Then, by means of next generation sequencing, we are going to get sequencing of DNA or RNA fragment. This gives qualitative and quantitative information. Next slide, please. Yeah. So far we have discussed about various techniques and various key attributes, various characteristics. So we are going to determine for the biopharmaceuticals. So for this purpose, multiple multiple techniques we are using. So that's why it is a very costly process for production of biopharmaceuticals, and a characterization of biopharmaceutical is a very costly affair. It is not a simple thing. It is, we need multiple analytical techniques. We need a multiple analytical techniques for characterization of key attributes. So those are what the different techniques we are going to use in quality control of biopharmaceuticals. Now coming to our objective, capillary electrophorese. Capillary electrophorese is a very widely used tool in the case of a biopharmaceutical analysis. CE, capillary electrophoresis, and CE coupled with the MS, CEMS are widely used techniques in the quality control of these biopharmaceuticals. So what is this capillary electrophoresis? So what is this capillary electrophoresis means? CE is a family of related technique. It is not a simple technique. It is a family of related technique. By means of a single instrumentation, we can run single instrumentation, we can run in different modes. That's why we are calling this as a family of related techniques. CE is a family of related techniques. And so we are performing electrophoresis, capillary electrophoresis in a capillaries, which are the diameter is ranging from 25 to 100 micrometer, 100 micrometer diameter capillaries, fused silica capillaries. We are going to use in here capillary electrophoresis, and the length of the capillary may be a 50 centimeter. So this is what the general dimensions of these capillaries, fused silica capillaries we are using. 50 centimeter capillary, and the diameter may vary from 25 to 100 micrometer. And by means of these capillaries, we are performing electrophoresis. So what is this capillary electrophoresis? So here in the capillary electrophoresis, what we are going to observe is there is a differential movement or migration of ions by attraction or repulsion in the electric field. So we are applying an electric field, and here there is a differential movement or a migration of ions towards a cathode and a anode. Cations will move towards a cathode, anions will move towards a anode, and there is a differential movement and there is a migration of ions. Thereby, separation of ions are going to be taking place in the electric field. So, in the capillary electrophoresis, how separations are going to be taking place? If you see the capillary electrophoresis basic instrumental setup, if you see the figure, there is a two wires will be there that we are naming as buffer and buffer. So there are two wires. That is what inlet wire and outlet wire. So both these inlet wire and outlet wire we are going to fill with buffer, that is what called background electrolyte. That is what we are simply calling as a background electrolyte. So for that we are filling in inlet wire and outlet wire. This inlet wire and outlet wire is connected through a silica capillary. So these two wires are connected by a silica capillary, and at the end of the silica capillary, if you observe a detector, there is a red colored portion, that is what called a detector. So at the end of a capillary, detector will be there. So this is what the basic setup. Inlet wire, outlet wire, and inlet wire and outlet wire are connected through a capillary, and at the end of the capillary, detector window will be there, and the distance from capillary inlet to the detector what called effective length, we are calling as the effective length. And here in the effective length, separations are going to be taking place. And in the case of a inlet wire, we are keeping anode, and in the case of a outlet wire, we are keeping a cathode. So when you apply a voltage, to this, so in the case of electric field, so cations will move towards a cathode. So that's why whatever the positive charged ions are there, those are passing through the detector and reaches the outlet wire, and our peak is going to be detected. So see, like this, separation of our molecules are going to be taking place. So the simple mechanism is simplest electrophoretic separations are based upon ion charge and size. Basing upon their ion charge and size, the separations are going to be taking place. Next slide, please. So this is what the representation of the capillary electrophores is, and how the separation of ions molecules are going to be separated in the case of a capillary electrophoresis. So what we discussed, there is a two buffer wires, inlet wire and outlet wire, and a buffer is going to be filled in inlet wire and outlet wire. Those two buffers are connected by means of a capillary. So whenever you introduce the sample, our sample, see, these are what the mixture of analytes that are there in our sample, and basing upon their ionic size and charge, separations are going to be taking place. So see, here at the outlet wire, anode is there. So that's why anions are coming first, then neutrals are coming, then cations will come, and those are going to be detected in the capillary electrophoresis system. So like this, peaks are going to be generated in the capillary electrophoresis system. Next slide, please. So coming to this capillary electrophoresis, here the efficiency is the most important thing. So compared with the HPLC technique, other liquid chromatography techniques, capillary electrophoresis technique, we are calling it as a highly efficient technique. We are calling it as a highly efficient technique compared to the LC. So why it is highly efficient technique, capillary electrophoresis? So in the case of a gas chromatography and liquid chromatography, the flow of the mobile phase is like a hydrodynamic flow. The flow is a hydrodynamic flow that we are calling as a parabolic flow. So we are going to observe in the columns, parabolic flow we are going to observe in gas chromatography and liquid chromatography. In both these techniques, parabolic flow we are going to observe. But in the case of a capillary electrophoresis, in the case of a CE, we are going to observe electroosmotic flow. So the flow is electroosmotic flow. In the case of a LC or GC, the flow is a hydrodynamic flow. So here, if you observe the flow behavior, in the case of a electroosmotic flow, the flow is plug-like flow. The flow is plug-like flow. So due to this plug-like flow, we are going to get very better separations and very sharp peaks we are going to observe in the capillary electrophoresis because the compounds are well resolved, completely resolved, well resolved in the case of a capillary electrophoresis due to this plug-like flow, and so better separations are going to be observed in capillary electrophoresis. That's why we are calling this technique is a more efficient technique compared with the liquid chromatography or gas chromatography. Next slide, please. Yeah. So, what are the basic principles that are involved in capillary electrophoresis? What are the basic principles? So, here, two things we are going to observe in the capillary electrophoresis. Those are what called one is electroosmotic flow, and another one is electrophoretic mobility. These are what the two principles that are involved in capillary electrophoresis. That is, one is called electroosmotic flow and another one is called electrophoretic mobility. So what is this electroosmotic flow and electrophoretic mobility? Electroosmotic flow means, so here in the electrophoresis we are using a capillary, so fused silica capillary we are using, and the silica capillary contains a negatively charged selenol groups will be there. So here the capillary is a negative charge. So whenever we pass a buffer through a capillary, whatever the positive ions are there in the buffer, those are going to be encapsulated the buffer. And whenever you apply a voltage, there is a sudden movement of total our fluid towards the cathode, towards the detector side, towards the cathode. So that is what capillary electroosmotic flow. That means bulk movement of fluid through the capillary that we are going to observe in the capillary electrophoresis. So due to this capillary electrophoresis, all ions, irrespective of charge, those are passing through the detector. Those are going to be separated, passing through the detector, and reaches the outlet wire. So by means of this electroosmotic flow, that is what one basic principle, bulk movement of fluid through the capillary, and then electrophoretic mobility. So what is this electrophoretic mobility? This electrophoretic mobility is the inherent characteristic of our ions. So you know that cations will move towards the cathode and anions will move towards the anode. This is the inherent capability of the ions. So basing upon these two principles, electroosmotic flow and electrophoretic mobility, separations are going to be taking place in the capillary electrophoresis, and then peaks we are going to observe what we are observing in the slide. So basing upon the charge, if those are double charged, so those are easily passed. through the detector. So in the case of a normal polarity mode, so we are placing a cathode at the outlet wire. So due to that, cations will move fast. So double charged ions will come first. Then single charged positive charged ions will then come. Then positive charge and very large radius, those are coming later. So like that, then neutral analytes will come. Then anions will come. And in the case of anions, large sized anions will come first. Then small sized anions will come at the end. So like this, separation of our analytes are going to be, our biopharmaceuticals are going to be separated basing upon the charge in the case of a capillary electrophoresis system by using electroosmotic flow and the electrophoretic mobility. Next slide, please. Yeah, so we discussed that capillary electrophoresis is a family of related technique. It is not a single technique. So we can run single instrument in multiple modes. We can operate the single instrument in multiple modes. So those, what are those modes? So if you see those are what called capillary zone electrophoresis. That is what one type of mode, capillary zone electrophoresis. And then micellar electrokinetic chromatography, that is what called MEKC. And then CEC, that is what called capillary electrochromatography. This is what one mode. And then capillary gel electrophoresis. This is what one mode of capillary electrophoresis. And capillary isoelectric focusing. This is what another mode of CE. And then capillary isotachophoresis. This is what one mode. And finally, chiral capillary electrophoresis. So these are what the different modes of capillary electrophoresis. By using the same instrument, we can run that instrument in these different modes. So simply, first one is capillary zone electrophoresis. This is the simplest form of capillary electrophoresis. This we are using routinely in our biopharmaceutical analysis. And in the case of a second one, that is what called micellar electrokinetic chromatography. Here, in the case of a micellar electrokinetic chromatography, we are using SLS surfactant above CMC, above critical micellar concentration, that we are adding to the buffer or background electrolyte, and thereby separations we are going to perform. And this micellar electrokinetic chromatography is very widely used in metabolite analysis or bioanalysis. We are using very widely. And then capillary electrochromatography. And this capillary electrochromatography is we are calling as a hybrid technique. This is a hybrid of capillary electrophoresis and HPLC. This technique also widely used in pharmaceutical analysis. And then another important technique, that is what called capillary gel electrophoresis. This capillary gel electrophoresis, so here conventionally gel electrophoresis we are going to perform, but in the case of a capillary gel electrophoresis, we are performing electrophoresis, gel electrophoresis in the capillaries. In the capillaries, we are going to form use. So the capillary gel electrophoresis is very widely used technique in the biopharmaceutical analysis, like proteins, peptides, monoclonal antibodies, or oligonucleotides. In all these biopharmaceutical analysis, this is another widely used technique. We will see how we are going to use this in the analysis of biopharmaceuticals like monoclonal antibodies. And then another important technique widely we are using in biopharmaceutical analysis is capillary isoelectric focusing. So by means of this capillary isoelectric focusing, we are separating our components based upon the charge. So basing upon the charge, our compounds are separated, and so we can determine the charge heterogeneity by means of a capillary isoelectrophoresis. That also we will discuss in the future slides, how we are going to use capillary isoelectric focusing for the analysis of biopharmaceuticals like monoclonal antibodies. So especially these three techniques, capillary zone electrophoresis, capillary gel electrophoresis, and capillary isoelectric focusing. These three techniques are very widely used techniques in the biopharmaceutical characterization and biopharmaceutical quality control. And other techniques are capillary isotachophoresis. So here, basing upon the speed of analyte, we can separate our analytes in the electric field. So that is what one technique. And another technique is what called chiral capillary electrophoresis. So here, by means of a chiral capillary electrophoresis, we can perform chiral separations very simply by means of adding chiral selectral to the buffer. So simply we are adding chiral selectors to the buffer like cyclodextrins. We are adding to the buffer and we can perform a chiral capillary electrophoresis in a very simple way. But in the case of HPLC, if you see, if you want to perform a chiral separation, we need a chiral column. Chiral column. Those chiral columns are very costly. So it is very difficult to perform a chiral separation by means of a HPLC. But in the case of a capillary electrophoresis, simply by adding chiral selectors to the cyclodextrins to the background electrolyte, that is what called buffer, and then we can perform chiral separations by means of these chiral capillary electrophoresis technique. So these are what the different modes of CE. So among all these techniques, for our biopharmaceutical analysis, we are going to use capillary zone electrophoresis, capillary gel electrophoresis, and capillary isoelectric focusing we are going to use. Now coming to the capillary zone electrophoresis. So what is this capillary zone electrophoresis? This capillary zone electrophoresis is the simplest form of capillary electrophoresis, and we are calling this capillary zone electrophoresis also called free solution capillary electrophoresis, and it is the simplest form of CE. So far, whatever we discussed about a capillary electrophoresis, that is applied to a this simple form of CE. So by means of simple charge and size separation based upon the charge and size of our analytes, so our molecules are going to be separated. So we can determine heterogeneity in the case of our biopharmaceuticals by means of a simple capillary zone electrophoresis. But the basic thing that is required for capillary zone electrophoresis are homogeneity of the buffer solution what we are using. So we are going to use highly homogeneous buffer and we are going to apply a constant electric field. So throughout the capillary, so better separations and reproducible separation, and we are going to observe a precision in the case of a quality control. So this is what called capillary zone electrophoresis. Coming to the next slide, capillary gel electrophoresis. So this capillary gel electrophoresis, so what we are using in the case of this capillary gel electrophoresis is we are using a simple polymers like dextran, polyacrylamide, or polyethylene glycol, polyethylene oxide, simple polymers, polymeric gel substances we are using. Those are all water soluble. So we are packing those gels in the capillary. We are packing those gels in the capillary, and then here we are going to use a coated capillary. Here we are going to avoid a electroosmotic flow. Here only separation is basing upon electrophoretic mobility. So we are avoiding electroosmotic flow. For avoiding electroosmotic flow, we are coating the capillary by means of a polyamide coating, and then we are packing the capillary with the gels and by means of a simple. So here when you pack a gel in the capillary, it is going to form a entangled polymer network is formed. So entangled polymer network is formed like what we are observing in the figure. So like that entangled polymer network is formed, and then it allows separations basing upon charge to size. The molecules which are of less size, so those are going to be encountered least resistance and those are coming fast. So they are eluted fast, they are coming fast. So like that, basing upon the size and charge, separations of your molecules are going to be separated. So we are going to analyze our biopharmaceuticals by means of this capillary gel electrophoresis. Now, in the future slides, how we are going to use this capillary zone electrophoresis and capillary gel electrophoresis and capillary isoelectric focusing in the biopharmaceutical analysis we will discuss. So this is what called simple form of capillary gel electrophoresis. In the case of this capillary gel electrophoresis, we are commonly employed SDS gel, sodium dodecyl sulfate gel electrophoresis. Commonly, we are going to use so for determining the molecular weight of our proteins or peptides, we are determining. So this is what about how we are going to perform a capillary gel electrophoresis. Coming to the next slide, capillary isoelectric focusing. So here what we are going to do in capillary isoelectric focusing. So here also we are using capillary. So we are going to use a coated capillary here in the case of capillary isoelectric focusing. And so here in the case of an inlet vial, so we are going to fill orthophosphoric acid. Most commonly, we are going to fill phosphoric acid in the inlet vial. And in the case of an outlet vial, we are going to fill a sodium hydroxide solution. But in the case of a routine capillary electrophoresis, inlet vial and outlet vial, we are filling with the same buffer solution. But in the case of capillary isoelectric focusing, we are filling inlet vial and outlet vial with inlet vial with acidic buffer like phosphoric acid, outlet vial with basic buffer, that is what called a sodium hydroxide solution. We are going to fill, and then we are going to add ampholites. Ampholites we are going to add, and whenever if you apply a voltage, whenever if you apply a voltage, what will happen in the case of a capillary isoelectric focusing is a pH gradient is going to be established. Different pH zones are going to be established in the capillary. So whenever if you introduce your sample into the capillary, basing upon the charge separations are going to be taking place. So here how separations are going to be taking place is basing upon your isoelectric point of your protein or a peptide that is going to travel some distance in the capillary, and when the pH gradient is equal to the pi of your analyte, the movement stops. For that analyte, movement stops and the net charge is zero, and thereby there is no movement of this your analyte. So like that, basing upon the isoelectric points of your analytes, and due to different due to acidic buffer and basic buffer we are using in inlet and outlet vial, when you apply a voltage, a gradient, pH gradient is established. That means different pH zones are established, different pH gradients are established along the capillary. So different isoelectric point analytes are moved at different places, thereby their net charge is zero, and then their movement is going to be stopped, and then separation of your proteins or peptides or analytes are going to be taking place. And then after separation, we are moving the zones. We are moving the zones through the detector by means of a pressure, applying a pressure to the inlet of the capillary, or we are using a simply acetic acid. If you use through the inlet of the capillary, our formed zones are moved through the detector, and thereby like this different zones are formed, and those are going to be detected through the detector window. So like that we are going to separate our molecules basing upon the isoelectric point by means of capillary isoelectric focusing. So like this, what we are observing in the figure, different zones in the bottom figure, different zones are formed. Those are what different isoelectric point of our analyte. So like that different zones formed, and then we are moving those zones with the help of a pressure or by applying a chemical, and we are detecting those through the detector window. So we are quantifying and we are identifying different isoforms of our biopharmaceuticals, different isoforms of monoclonal antibodies. We are determining by means of this capillary isoelectric focusing. Next slide, please. Then applications of these CE applications in upstream and downstream biopharmaceutical process monitoring. So this process monitoring of biopharmaceuticals. Generally, these biopharmaceuticals we are produced by means of a living organisms like fungus, yeast, bacteria like E. coli, and then mammalian cells. The most commonly Chinese hamster ovary cells were commonly used. So these biopharmaceuticals, proteins, peptides, monoclonal antibodies, we are producing by these living organisms. And so we are dividing the production process into two steps. That is what called upstream processing and a downstream processing. Biopharmaceutical development, we are dividing into two processes. That is what called upstream processing and a downstream processing. So what is this upstream processing and a downstream processing? If you see that in the case of upstream processing, that is nothing but a production of our biopharmaceutical. Upstream processing is called as production of biopharmaceutical, and a downstream processing is called as purification of our required product, and the development of a stable formulation. That is what the upstream process and the downstream process. Upstream process is production process, production of our POI, our required product of interest, and the downstream process means purification of a produced biopharmaceutical. That is what called downstream process, and the development of a stable formulation is also a downstream process that we are calling as USP and DSP. Next slide, please. So in the case of this USP and DSP, so our objective is we are going to produce a stable, safe, and efficacious drug product, efficacious biopharmaceutical. So stable biopharmaceutical and safe biopharmaceutical and efficacious biopharmaceutical or biosimilar we need to produce. So for this, during the production process, each and every step, we are monitoring the protein expression. Protein expression is going to be there. So we are going to optimize the protein or peptide expression by means of a capillary electrophoresis tool. Each and every step of the production process, we are going to optimize cell culture conditions. So different pH, different amino acid, different nutrients, we are going to add to the cell culture conditions. In the cell culture conditions, each and every step, we are monitoring the output. We are going to analyze the yield of our product. So for the quantification of our product, we are going to use a capillary electrophoresis in each and every step. So if you see this biopharmaceutical production, the production is very complex and the matrix is a very complex. It is not that much easy to analyze each and every step in the biopharmaceutical production because the matrix is a very complex. This is a cell harvest or a cell culture media we are going to analyze. So if you see the CE is the most widely used technique, and this is having a number of benefits compared to other techniques. If you see those, it has ability to handle high complex matrices. So in the case of a production of biopharmaceuticals, the matrix is very complex. So such a complex matrix we are handled by means of a CE. And then another advantage of CE is it has a high resolving power, very sharp peaks and high resolving power between components. We are going to use widely this CE technique. And another advantages, benefits of these CE techniques are very little sample required, very nanoliters of sample, very a small sample amount is required. Nanoliters is enough to analyze by CE. And then the background electrolyte or buffers, whatever we are using also in mls only, few ml, 2 ml is enough. So that's why the CE technique is very widely used in the biopharmaceutical production monitoring and quality control analysis. So now the latest developments are microchip based capillary electrophoresis systems. People have developed chip based CE systems also. So microchip CE. So microchip CE offers advantages like PET, process analytical technology tool. That means we can monitor our biopharmaceutical production online and real time monitoring we are performing by means of this microchip CE techniques we are going to use for online monitoring. That is what called a PAT analytical tool, process analytical tool. So that is what how this CE technique we are using in upstream processing and downstream processing in the case of a biopharmaceutical production. If you see the biopharmaceutical production, protein expression is different in different stages. So each and every stage we are going to monitor the protein expression. So we are going to purify our sample and analyze by capillary electrophoresis. So for this, very simple preparation is enough and very nanoliters of sample is enough. So we can easily analyze by upstream processing and downstream processing by means of this CE. Please, next slide, please. Yes. So, what are the critical quality attributes we are monitoring in upstream and downstream processing of this biopharmaceutical production? If you see the critical quality attributes, or what are the characteristics, critical quality characteristics or attributes we are analyzing is all characteristics that are influencing the safety of our product, efficacy, and quality of product we are considered as critical quality attributes. These are whatever the parameters that are influencing the safety, efficacy, and quality of a product, we are calling it as a critical quality attributes. And in order to control these critical quality attributes, we are characterize critical quality attributes during the production. It is very much essential to control the critical quality attributes. So, what are these critical quality attributes in the biopharmaceutical production? Are one is size heterogeneity. You know that biopharmaceuticals are very big molecules. So, size heterogeneity. So, we are going to monitor during the biopharmaceutical production. And then those are having different charges. So, charge heterogeneity we are going to observe, charge heterogeneity we are going to monitor, and then glycosylation. So these are what the three critical quality attributes of the biopharmaceutical production in the case of a proteins or peptides or a monoclonal antibodies or a vaccines. So these we are going to observe size heterogeneity, charge heterogeneity, and glycosylation. And if you see how these we are going to monitor by means of a capillary electrophoresis techniques. Next slide, please. Yeah, size heterogeneity. So, in the case of a biopharmaceuticals, the size heterogeneity is a very, very important thing. So, here the size heterogeneity comprises a product related aggregates are going to be formed. In the production of this, during the stage, are going to identify and quantify these aggregates. These are nothing but high molecular weight species. So high molecular weight species are going to be formed. That is what called aggregates. And also fragments are also going to be formed from our biopharmaceutical products. So those are what called low molecular weight species. So we are going to separate from our molecule whether it is intact or is there any aggregates are formed, or fragments are formed in your biopharmaceutical production, we are going to observe continuously for batch to batch consistency. So, for this purpose, for the analysis of these aggregates or high molecular weight species or fragments or low molecular weight species, we are going to use capillary electrophoresis tool. So, what is the capillary electrophoresis tool? We are going to use is capillary gel electrophoresis. Because in the capillary gel electrophoresis, separations are basing upon the size to charge. So, due to this size to charge, better separations and well resolved separations, even though there is a very narrow size differences, very small size differences also we can monitor by means of a capillary gel electrophoresis. Now coming to the efficacy of our therapeutic proteins, the efficacy of our therapeutic proteins is depending upon the structure, core structure of our biopharmaceutical. If the structure is altered, the efficacy of our biopharmaceutical product is going to be altered. If you see that due to the formation of these aggregates, aggregation of therapeutic proteins can cause in loss in function. So the function is going to be lost, and there may be a chance of due to these aggregates, immunogenic responses we are going to observe, and also adverse effects are also going to be observed due to these aggregates. This ranges from protein neutralization to anaphylactic shock we are going to observe due to the aggregates. So that's why it is very important during the biopharmaceutical production. You have to monitor the aggregates, we have to monitor the fragments. So all these high molecular weight and low molecular weight fragments we are separating by means of a CGE. That is what called sodium dodecyl sulfate capillary gel electrophoresis we are going to use commonly. Next slide, please. Yeah, this is what the simple example of a IgG antibody, immunoglobulin G antibody, and how these high molecular weight fragments and low molecular weight fragments and uh, aggregates are separated by means of a SDS-CGE. SDS-CGE, we are telling that sodium dodecyl sulfate capillary gel electrophoresis. So here what we are going to do is first we have to denature our protein or denature our biopharmaceutical with the help of a SDS. With the help of a SDS, we are going to denature our protein. So if you denature our protein, our intact primary structure is as it is. The intact primary structure is as it is. Two-dimensional or three-dimensional structures are going to be lost, but intact structure will be there. And then we are going to give a uniform charge to our biopharmaceutical by means of treating our biopharmaceutical with sodium dodecyl sulfate. That's why uniform negative charge we are giving to all our biopharmaceuticals by treating with SDS. That's why we are most commonly performing SDS capillary gel electrophoresis widely. And again, this SDS capillary gel electrophoresis we are performing in two modes. That is what called reduced mode and non-reduced mode. So in the case of a reduced mode, we are using mercaptoethanol so as a reducing agent. So thereby disulfide bonds will be broken and we can observe different fragments in the case of a reduced mode. And in the case of a non-reduced mode, we can analyze our intact monoclonal antibody or intact biopharmaceutical in the case of a non-reduced immunoglobulin. If you see the figure, in the figure, the top one is a reduced immunoglobulin G electropherogram, and the down one is a non-reduced immunoglobulin G electropherogram. And the first numbered peak is that is a internal standard of 10 kilodalton size, and the second one is light chain of a monoclonal antibody. That is a fragment of monoclonal antibody. So that is a second peak. And the third numbered peak is a non-glycosylated heavy chain. So here another important thing in biopharmaceuticals is glycosylation is very important. Glycosylation of biopharmaceutical is very important basing upon the glycosylation is only our efficacy of biopharmaceutical will depend. If your biopharmaceutical is a non-glycosylation portion, if your biopharmaceutical contains more amount of non-glycosylated biopharmaceutical, your efficacy of our product is very, very less. That's why we are going to monitor non-glycosylated portion of our monoclonal antibody or other biopharmaceuticals. So if you see the diagram, in the reduced diagram, electropherogram, the third number peak is non-glycosylated heavy chain, and the fourth peak is heavy chain. So that means the non-glycosylated heavy chain is clearly separated from the heavy chain. So there is a baseline separation between non-glycosylated chain and glycosylated chain. So due to this advantage, we are going to perform capillary gel electrophoresis to identify, to quantify the non-glycosylated portions of our biopharmaceuticals. Thereby, we can see our produced biopharmaceutical is having effectiveness or not, or produced biopharmaceutical is having batch to batch consistency for the quality control release. It is very useful. Coming to the non-reduced immunoglobulin G monoclonal antibodies electropherogram, if you see the six peak, the six peak is a two heavy chains are there, two heavy chains of monoclonal antibody is there. And in the case of a seventh peak, two heavy chains and one light chain is there. And coming to the eighth peak, the eighth peak is a non-glycosylated heavy chain. And the ninth peak is immunoglobulin G monomer. So if you see that 8 and 9 peaks, the 8 and 9 peaks are our our intact antibody and non-glycosylated antibody. So in this case of a non-reduced electropherogram, the baseline resolution between non-glycosylation portion of your non-glycosylated biopharmaceutical and simple intact monoclonal antibody is poor. The resolution is poor. That's why we can identify non-glycosylation portion in the non-reduced form, but in the case of a reduced form, we can easily resolve non-glycosylation portion from glycosylation portion of our monoclonal antibody, and thereby we can identify and quantify the glycosylated portion and the non-glycosylation portion, and as well as basing upon our sizes, separation of biopharmaceuticals we are going to perform by means of a capillary gel electrophoresis. That is what called commonly SDS-CGE. SDS-CGE. So this is what the benefit of SDS-CGE. So by means of this SDS-CGE, we can determine size heterogeneity, what are the different sizes that are there in our biopharmaceutical, and then glycosylated portion and non-glycosylation portion also we are going to identify by means of this. Next slide, please. And then charge heterogeneity. So the charge heterogeneity that is going to be generated through post-translational modifications. By means of post-translational modifications, charge heterogeneity will be occurred. If you see that post-translational modifications could result in more acidic species by means of sialation, deamidation, or glycation, or more basic isoforms are also going to be generated by means of succinimide formation or incomplete removal of C-terminal lysine in our monoclonal antibodies or peptides. So, depending upon the position of this PTM, it could affect the antigen binding affinity. So, basing upon this charge heterogeneity, our binding affinity is also going to be altered for the biopharmaceutical. Thereby, potency is varied and the secondary structure will be disturbed, and there may be a chance of aggregate formation due to these post-translational modifications. So it is very important to identify the charge variants in our biopharmaceutical. So the charge variants we are going to be determined by means of a technique called capillary isoelectric focusing. The technique is what called capillary isoelectric focusing. So this technique now, the advanced technique is also there. That is what called imaging capillary isoelectric focusing. So we can run capillary isoelectric focusing like a simple capillary isoelectric focusing. So, and another one is image capillary isoelectric focusing. So what is the difference between this image capillary isoelectric focusing and the simple capillary isoelectric focusing is after formation of zones. We already discussed in the capillary isoelectric focusing slide. So there is a generation of or a formation of zones basing upon the isoelectric point of our analyte. Different zones are formed. Those zones are passed through the detector window by means of a pressure or by means of a chemical in the case of a capillary isoelectric focusing. But in the case of a imaged capillary isoelectric focusing, we are not using any physical or chemical technique to pass those zones through the detector window. Simply, we can take an image of the capillary. We can take the image of the capillary and we can know the positions of the our zones in the capillary window. So thereby we can determine the isoelectric point. We can determine the isoforms of the charged isoforms, charged variants of our biopharmaceuticals by means of capillary isoelectric focusing. Next slide, please. So this is what a one example of a monoclonal antibody separation of capillary different isoforms of monoclonal antibody. This is the analysis of monoclonal antibody, one monoclonal antibody. So here, if you observe the electropherogram, uh, uh, so there are three different zones we are observing. One is main isoform group, another one is basic isoform group, and another one is acidic isoform group. So these are what the different isoforms apart from our main isoform group. These are what the different isoforms we observed in the monoclonal antibody, and then basing upon this capillary isoelectric for isoelectric focusing analysis, we can know how much percentage of basic isoform, how much percentage of acidic isoforms are there in our monoclonal antibody. So like that, basing upon the percentages of acidic and basic isoforms, we can know whether our monoclonal antibody is of having batch to batch consistency, or is there any variations of these acidic and basic isoforms in batch to batch? These are more. So what will happen? It may happen we are going to analyze bioanalysis and we can test due to the increase in acidic isoforms or increase in basic isoforms, there may be a chance of less efficiency of biopharmaceutical. So due to these reasons, we want to analyze charge variations of our monoclonal antibodies or proteins or peptides of biopharmaceuticals. So we can quantify those, how much percentage of these peaks are there, basic isoforms and acidic isoforms are there, and then we can release the batches of biopharmaceuticals in the industry by means of this quality control testing. Next slide, please. And already we have discussed in the capillary gel electrophoresis only glycosylation. This is another important critical quality attribute of biopharmaceuticals. So generally glycosylation, there may be N glycosylation and O glycosylation. And among this N and O, N glycosylation is the most common form. And we can, in the majority of therapeutic proteins, we can observe N glycosylation. So what is the importance of this glycosylation? So how it is going to influence various factors of our biopharmaceutical is, So this glycosylation pattern of our biopharmaceutical will influence the biological activity of your biopharmaceutical. Stability will be altered basing upon this glycosylation. And pharmacokinetics and pharmacodynamics are also going to be altered basing upon the glycosylation of your biopharmaceutical. And then, basing upon this glycosylation, immunogenicity is going to be altered. So, the safety and potency of our biopharmaceutical is highly depending upon the glycosylation pattern of our biopharmaceutical. So this glycosylation, so that's why we want to determine the glycosylation amount of our biopharmaceutical. So for determining this, so we are going to determine the glycan content of your biopharmaceutical. So for releasing of biopharmaceutical or for releasing of monoclonal antibody into the market before the batch analysis in the quality control test, we need to perform the glycan content of your monoclonal antibody or other protein or peptide formulation. So for this glycan content, how we are going to determine, for example, for a monoclonal antibody, how we are going to perform glycan content analysis. So for this glycan content analysis, what we are going to do is, first we have to release these oligosaccharides or glycans from our monoclonal antibody. So the release of N-linked oligosaccharides by means of a enzyme called peptide N-glycosidase F, PNGase F. This is what the common enzyme we are treating with monoclonal antibodies or biopharmaceuticals to release the glycan content of the biopharmaceuticals. And then you know that biopharm sugar moieties, those are not having any absorption by means of UV. So what we are going to do is, so we have to label these charged sugar molecules, charged analytes, with the help of a suitable labeling reagents. And then we are going to perform simple capillary zone electrophoresis. And the detector we are going to use is laser induced fluorescence detector, LIF. So capillary zone electrophoresis, we are performing it to UV detector. And in the case of a biopharmaceuticals, another mode widely, another detector widely used is laser induced fluorescence detector. So by means of this laser induced fluorescence detector, we can quantify, we can identify, or we can quantify the glycan content that is there in our monoclonal antibody or in the biopharmaceutical. Next slide, please. This is what the one simple model electropherogram of a monoclonal antibody glycan content analysis. These are G0, G1, G2. These are what the different glycan fragments that we observe from a monoclonal antibody. So one diagram A is simple our monoclonal antibody, what are the different glycan fragments we observe, and B is the blank that is spiked with different in the market commercially available glycan standards are available. So in the blank we spiked with those are glycan standards. I can and we can compare those glycan standards with our glycan moieties we observed in our monoclonal antibody, and thereby we can compare the glycans and we can quantify the glycans. Thereby we can release the product into the market by means of this glycan content. Next slide, please. Yeah, these are what the different critical quality attributes: charge heterogeneity, size heterogeneity, and glycosylation. So all these critical quality attributes we are measuring with the help of a capillary electrophoresis technique. So here we are using CE, and along with CE, CEMS, capillary electrophoresis coupled with the MS. And in the case of a MS, the most common ionization mode we are going to use is a electrospray ionization technique. And by means of this CZEMS technique, we can identify the these glycan contents by means of this CE and MS techniques. And if you see in future, so we are already observing a microchip based CE and CEMS analysis. So this CE and CEMS analysis is useful as a online tool. online tool, or that we are calling it as a PAT tool, process analytical tool. So these CE and CEMS techniques are widely used in biopharmaceutical quality control and biopharmaceutical product development, and these are highly useful for batch release of our biopharmaceutical products in the pharmaceutical industry. So these are what the references, some of the references. And then finally, thank you.
Thank you, sir, for your wonderful talk on this one of the technologies that is academicians. I can say that students are very few students are aware about such kind of technologies that is nowadays utilized in the industry sectors, especially for this kind of high molecular weight molecules.
Thank you, sir.
And sir, the presentation was really wonderful. That was very understandable by the students or the delegates who have participated. The way you have presented the diagrams of how the ions are moving and how it is detected, was a really nice one, sir.
Thank you, sir. Thank you so much.
Any question is there from the audience or participants?
Good afternoon, sir. Thank
Good
you for your
afternoon.
elaborative presentation.
Thank
Myself,
you.
myself, Mridulata from 4th B Pharmacy.
Okay.
Sir, my question
Yes.
is: you have delivered so many analytical techniques with various attributes. Which
Yes.
of these analytical methods are most commonly adopted in industries for routine characterization of biologies?
Yes. 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 These are what the different techniques we are going to use in the quality control of or in the production of biopharmaceuticals. That's why the cost of a biopharmaceutical or biologic or biosimilar is very, very high, because we need to use multiple techniques, and those are our very costly techniques, and the expertise of those techniques is also very poor in India. So that's why these are very costly and costly process. So that's why the cost of the product is also very high. So it is very much essential to use all those quality control techniques to release a biopharmaceutical into the market.
Thank you, sir, for your elaborative session.
Thank you.
Thank you, Dr. Vamsi Krishna, for educating us on analytical technique used in the industry,
Yes.
and we are really thankful to you for
Thank you,
this,
sir.
for imparting knowledge to us.
Yes.
Thank you very much.
Thank you, sir. Thank you so much.
Thank you very much, sir, for
Thank you.
addressing the queries with an enlightening and informative presentation. Your
Yes.
practical insights and industry experience have provided valuable perspective for all the participants. As a token of appreciation, we would like to present a certificate of appreciation to our esteemed speaker,
Okay.
Vamsi Krishna, sir.
Yes. Yes.
Thank you, sir.
Thank you, Amma. Thank you very much.
With this, we come to the end of today's scientific session. On behalf of the organizing committee, I would like to express our sincere gratitude to our esteemed distinguished speakers, respected chairpersons, and organizing committee members, and all the participants for your valuable presence and active participation. We look forward to your continued participation in tomorrow's offline scientific sessions also. Tomorrow's offline scientific sessions are by Professor S. Shanmuga Sundaram, sir, Wales University, Chennai, Tamil Nadu. followed by next scientific session by Professor Vittal V. Chopde, PES Modern College of Pharmacy, Pune, Maharashtra, followed by valedictory function. Thank you once again. Have a wonderful day ahead.
Okay, thank you.
Thank you all.
Thank you.
Thank you very much, sir. We will meet in the tomorrow's session. Thank you, one and all.